We're all very familiar with blood because we all have seen a cut where we may have some bleeding come out, or maybe we've gone in to the doctor's office for a blood draw. So today we're going to talk about all of the different components of blood, as well as some of those functions that the blood is going to provide to help us maintain homeostasis. So when we're looking at blood, this is actually a specialized connective tissue. You've seen it on the microscope in lab, and a single drop of whole blood will contain about 260 million red blood cells, which is phenomenal to even think that there's 260 million cells within that one drop of blood. When you're looking at the microscope slides, what you will also notice is you will see far more red blood cells than you will see white blood cells. So we have about 1,000 red blood cells for each white blood cell. That's why it's much easier to see the red blood cells on the slide, much easier to find them, whereas you will have to go actually searching for those white blood cells because they are not as numerous. There are multiple functions of blood. This includes transporting those dissolved gases like oxygen and carbon dioxide, providing nutrients, hormones, and then moving metabolic waste. We are going to also be able to help regulate the pH with the blood, the ion composition of the interstitial fluids, restricting fluid losses at injury sites, so making sure that the blood clots whenever there is a cut or an open wound, defending against toxins and pathogens, so this will be involved in immunity, and then of course stabilizing body temperature as well. Blood is normally about 38 degrees Celsius, so this is 100.4 degrees Fahrenheit. Normal body temperature is 98.6, so as you can see, blood is a little bit warmer. It is highly viscous and then slightly alkaline, so please make sure that you do remember and never forget that the pH of blood is between 7.35 and 7.45. This is going to become very important as you get into clinical practice, as you evaluate maybe some arterial blood gases. When we talk about the pH balance, this will be important as well in remembering that blood is not neutral. It is not a 7.0. So we are looking at 7.35 to 7.45 for that slightly alkaline characteristic of blood. In regards to volume, this is about 7% of the body weight. And average, you can think about somewhere between 5 to 6 liters of blood in a person. So males are going to have a higher amount than females. So I put the little big reds down there so that you can remember in males, so that's a 2 liter of big red, so we've got about 6 liters, whereas in females, we've got about 5 liters. Remember our cardiac output equals heart rate times stroke volume example, where my son and I did the little example of pouring the tomato juice into the bowl. So we were talking about how much blood can come out of the heart and then go into the systemic circulation in that particular example. So this is just here as another kind of visual reminder to help you remember about the average amount of liters in males versus females. When we're looking at whole blood, we have plasma, which is going to be the fluid component of blood, and then we also have what we call formed elements, which will be cells and cell fragments. If we do fractionation, that means that we're basically going to separate that whole blood into those different components. You can see here that whenever you do a blood draw, they're going to put it in a little centrifuge, which is going to spin really fast, which will separate out the different components of the whole blood. The plasma is going to come up towards the top of the tube because it is not as dense as those formed elements. So the plasma is mostly going to be water. We'll talk about that in a minute, but the plasma is 55% of the whole blood, whereas the erythrocytes, which is our red blood cells, are going to be 45%. You can see there that the leukocytes, which is going to be your white blood cells, and then your platelets, are less than 1% of the whole blood. So we've not got a lot of white blood cells. Remember, we have far more red blood cells than we do white blood cells. So the buffy coat will be that layer between the plasma and the erythrocytes that will consist of those white blood cells and platelets. Altogether, we will call the buffy coat with the erythrocytes the portion that has the formed elements in it because that's made up of the cells and then the cell fragments. Whenever you're looking at hematocrit, you can see there hematocrit is the percentage of blood volume that is actually red blood cells. So males is going to be about 47%, females about 42%, both of those plus or minus 5% when we're looking at that red blood cell percentage for your blood volume. Again, plasma is 55% of the blood volume, and more than 90% of plasma is water. So I've talked to you guys over and over and over again about the importance of drinking water, making sure that you're constantly drinking half of your body weight in ounces. As you can see here, your plasma is going to be 90% water. You have to have water in order for your cells to do what they need to do in order for them to metabolically work and maintain homeostasis. And then you've also got a lot of water within your blood plasma as well. So make sure that you are drinking your water. Your body needs it. You cannot go without it. And we will see changes in blood labs whenever there is dehydration as an issue. Similar in composition to the interstitial fluid is this plasma, and you can see here this is because water ions and then the small solutes are exchanged across those capillary walls. If we're looking at this picture here, you can see that within plasma we do have plasma proteins, 7%, we have other solutes, 1%, and then water, 92%. So we've got to have this great amount of water in order to help us create this plasma that our body is going to need. Again, in regards to formed elements, we've got about 45% of that for the whole blood concentration. Majority of that, you can see 99.9% of those formed elements will be your red blood cells. Very little on the white blood cells and the platelets. Again, you can see there a reference to hematocrit, and this can also be called packed cell volume, PCV. So those mean the same thing, whether you're saying hematocrit or whether you're saying PCV. The sex difference between males and females is going to be because of androgens. So androgens will actually stimulate red blood cell production. Estrogens do not. So males are going to have a higher hematocrit than females because they have a higher proportion of androgens within their system. Okay? The other thing I want to draw your attention to is down in the right-hand corner where you can see that it says formed elements are produced through the process of hemopoiesis, also called hematopoiesis. We'll be talking about that more here in just a few slides, but hemopoiesis is how we're going to actually create these formed elements. So, as I said, we're going to talk about that later on, but there's going to be a lot of new vocabulary as we go through this particular PowerPoint, so I want to make sure that you are able to keep all of these new words separated and you know what each one is referring to. Although plasma is mostly water, remember about 7% of it is proteins. So let's talk about the different types of proteins that you would find in plasma. The predominant one is called albumin, and albumin is very important because not only does this help transport different things like fatty acids and hormones, this also helps contribute to plasma osmolarity. That basically means the measure of the hydration status. So when we're looking at plasma, of course, remember a lot of this is water. Albumin will help keep water within the blood vessel so that it doesn't leak out of the vessel and give us all of these pockets of edema all over the body. So albumin is very important in not only moving things around the bloodstream, but also maintaining that water volume within the blood vessels themselves. The globulins is another important plasma protein, so we're going to talk about these later on. These will consist of the antibodies, sometimes known as Igs, or Ig basically means immunoglobulin. Fibrinogen is a very small percentage, but this is a soluble protein that will function in clotting. It can be converted to the insoluble fibrin, and whenever you have conversion of fibrinogen to fibrin, it will leave the serum in the blood sample. Other plasma proteins are just at 1%, and this will be varying concentrations of enzymes and hormones. So although that albumin will be talked about the most, just keep in mind it is not the only plasma protein. More than 90% of these proteins are going to be made in the liver, so it's very important that you have a healthy liver in order to help create these plasma proteins. As you can see here, if your liver is not healthy and functioning well, you are going to disturb a number of homeostatic mechanisms within the body. So liver tests will be done on blood labs, but there's a little caveat to those. We'll talk about that later on. Antibodies will be made by plasma cells, and then you have peptide hormones that are made by endocrine organs. Again, our formed elements include the RBCs, WBCs, and the platelets. Platelets are not actually whole cells themselves, they are just cell fragments. You will see that later on in a little picture. And remember that word hemopoiesis or hematopoiesis is the process of producing those formed elements. Here are our different formed elements. As you can see here, the platelets are those small little cell fragments. They are going to be very necessary for clotting, so make sure you do circle that. And you know that their primary function is to help you prevent you from bleeding out. White blood cells, you have 5 different types that you see listed there. We'll talk through each one here in a little bit. These are all about immunity. So make sure that you write that word immunity or circle body's defense mechanisms right there in that picture so that you know the primary role of those white blood cells. And then red blood cells, it's all about transporting oxygen in the blood. So that is the primary function for our erythrocytes, is making sure that we can have oxygen transported through the various tissues of the body. We're going to talk about the red blood cells first, then the white blood cells, and then lastly we will go through the platelets. So again, you have about 99.9% of your formed elements as erythrocytes. This is going to make up about 1/3 of your body cells, these red blood cells do. So there's approximately 25 trillion red blood cells, which is a lot, and we have about 75 trillion cells inside of our body. The very important component of red blood cells is going to be the hemoglobin. So make sure you do not forget this word, because hemoglobin is going to be not only the red pigment that will give blood its color, but most importantly the one that is going to bind and transport oxygen and carbon dioxide. So we'll be talking about hemoglobin and its role in maintaining homeostasis. Just do not forget that hemoglobin is within the red blood cells to help transport those gases throughout your system. When it comes to our RBC count, this is the number of red blood cells per microliter of whole blood. Again, males are going to be higher than females. So reference ranges are going to vary depending upon the lab that you choose, but this will be somewhere between 4.5 to 6.3 million for an adult male and 4.2 to 5.5 million for an adult female. Again, hematocrit, when we measure that, this can also be called the packed cell volume, and we're looking at the percentage of the formed elements within the blood, higher in males than it is in females. When this test is run, it's important to note that this is not a specific diagnostic test. It just lets you know if you need other studies, because if you are high or low, there are a number of different reasons why your hematocrit could be high or low, so you need to be a detective and dig a little bit further to figure out what's really going on. So as you can see the example there, this hematocrit is going to increase with dehydration due to the decreased plasma volume. Remember, plasma volume is 90% water. So if you're low in water, then that means that the percentage of the formed elements in the blood is going to be higher because your water is too low. Whereas if you have an internal bleed that you're unaware of, then your hematocrit is going to be low, because obviously your red blood cells are not going to be within your bloodstream. They're coming out into the tissues with that internal bleed. So Those are just two examples of things that could cause an abnormality in hematocrit, but again, you have to go further and run other tests to figure out what is going on. When we're looking at the structure of the red blood cells, these are very small and these do not look like other cells. We call these biconcave disc because there is an impression on both sides of the cell. So you can see there in the picture there's a little dip, and that's going to happen not just on one side but on both sides. So you have that very thin center and then that thick outer margin. This is why whenever you're looking at the slides in lab, you will notice sometimes that the red blood cells, they look kind of hollow in the center. It's not that they're hollow, it's just that the stain is not going to be picked up on that biconcave shape within the center as much as it will on that thicker outer margin. So that's why it looks like that on the slide. You can see here that they are very small if we're comparing it to something like a piece of hair. The red blood cell, you can see, is significantly smaller than that hair. Whenever we're looking at the purpose of this shape, the reason is because we want a large surface area to volume ratio. Whenever we have this larger surface area to volume ratio, this will allow there to be a quick response to changes within releasing oxygen. So very important that we're able to oxygenate quickly, and the shape of the red blood cell will help allow that to happen. The other thing that the shape will do will allow it to form these stacks of red blood cells called rouleaux, and this will allow for there to be smooth blood flow through even narrow blood vessels. So we don't want turbulent, we don't want backed up flow, we don't want things, hitting the wall and causing problems with the vessel. So we want to be able to create these kind of stacks that will allow there to be a continuous smooth blood flow through these vessels. The other thing is whenever it gets to the small tiny capillaries, which are all the way in the periphery, even the small tiny ones like in your pinky finger, the blood cell can actually bend, and if it bends, it can flex and go through the smallest of capillaries without creating any issues. So this biconcave shape allows it to be flexible and to move through even very small spaces. Whenever we're looking at the red blood cell, it's important that you know that they are anucleate. There is not a nucleus within the red blood cells, and they lack mitochondria and ribosomes. For these reasons, they are unable to divide. They cannot make their own proteins, and they cannot repair damage. So therefore, red blood cells are only going to live for about 4 months, or about 120 days. So do not forget that number, 120 days, because they are not able to do those things that you see out listed in red. Therefore, we're going to constantly need new supplies of red blood cells, and we will get them every single day. When it comes to sickle cell disease, this is a pathology in the actual shape of the cell itself. So you can see the picture on the right. What we should have is a biconcave disc, but instead of having that biconcave disc, it looks like this little sickle. And so what's happening is now we don't have the ability to kind of create those stacks of rouleaux, and we don't have the ability to fold and bend and get through the small little capillaries. So this shape has been altered, which means our blood flow is going to be altered, which means our homeostasis is going to be altered as well. So inside of the red blood cells, the hemoglobin molecule, remember the one that will carry the oxygen, is going to have a malformation. And so this malformation is what is disrupting the shape of the red blood cell. In sickle cell disease, you get 2 copies of a gene that's called hemoglobin S that are inherited, one from each parent. In this gene, actually, the mutation evolved in Africa, where this sickle shape actually prevented the malarial parasite from continuing to live on in the blood. So it did offer an advantage against malaria whenever you lived in an area where this was really high. However, if you're not living in Africa anymore, but your parents have these traits that get passed on to you, now there's really no competitive advantage if you're not really struggling with a malaria-infested region and you're just living somewhere where you don't have to worry about that. It is going to still cause these problems. So you will have anemia that will frequently accompany sickle cell disease because those cells only live for about 10 to 20 days. So remember, 120 days for normal red blood cells. 10 to 20 days for sickled red blood cells is going to be problematic for a number of different reasons, but the bone marrow is not going to be able to keep up with their replacement. So even though we get new cells every single day, the fact that these are declining at a much more rapid rate is going to be problematic for that person. So of course, proper nutrition, because we'll talk about what is necessary in order to help create red blood cells, so proper nutrition and hydration. Obviously, you just heard how important water is in regards to the blood. Both of those are going to be incredibly important, and there have been advances in stem cell therapy that have proven to be helpful for people with sickle cell disease as well. All right, so again, that hemoglobin is that protein in the red blood cells that will transport the respiratory gases. Normal hemoglobin in an adult male is 14 to 18 grams per deciliter. As you can see in a female, 12 to 16. So males are always going to have higher readings on their blood counts than females are, because remember, they have more red blood cells, so therefore they're also going to have more hemoglobin. When we're looking at hemoglobin, this is a protein, and so we will call this a quaternary structure, quat meaning 4. So you have 4 globular protein subunits. You have 2 alpha chains, which you can see in purple, and 2 beta chains, which you can see in blue. So each comes with 1 molecule of heme, which you see in red. It's the little red circle. And each heme contains 1 iron ion. So you see the arrow pointing to the iron within the center of the heme. The iron is going to interact with the oxygen to form what is called oxyhemoglobin. So if you have iron bound to your heme, then we would say that is in the form of oxyhemoglobin. Again, if we have tissues that need oxygen, then that red blood cell is going to have to give up its oxygen and put it over into a tissue. Now this can be very easily done, and when this happens, we would then say that now we are at a form called deoxyhemoglobin. Deoxyhemoglobin means that there is just no longer oxygen that is bound to that heme molecule. Okay, so any disorder that would cause hypoxemia can generate sufficient deoxyhemoglobin in the blood, leaving the heart to produce central cyanosis. Central cyanosis is going to be caused by reduced arterial oxygen saturation. Not enough oxygen within the blood. Central cyanosis is, of course, going to be associated with life-threatening illnesses such as cardiac, metabolic, neurologic, infectious, etc., etc. All these different disorders, because we have to have good oxygen saturation. So hypoxemia, low oxygen, okay? Too much deoxyhemoglobin, we need more oxygen. So hypoxemia is basically defined as an arterial deoxyhemoglobin level greater than 2.38 grams per deciliter, corresponding to oxygen saturation of less than or equal to 80%, and an oxygen percentage of less than or equal to 45 millimeters of mercury in patients with normal amounts of hemoglobin. So when it comes to our oxygen saturation, we want 95% and above. Okay, that PO2 is the partial pressure of oxygen. We'll talk more about partial pressures of oxygen whenever we get into the respiratory system. So oxygen saturation less than 95%, we're going to start to get a little bit concerned. But as you can see here, at 80%, that is far too low and is going to be causing multiple life-threatening issues. When it comes to the babies in the womb, they have a different form of hemoglobin. So we would call this fetal hemoglobin. And this form has a greater affinity than adult hemoglobin, which basically means it can bind oxygen more readily and more easily than does an adult hemoglobin. So you can see here it says takes up oxygen from maternal blood at the placenta. The baby has the ability to take more from mom, sort of like stealing it away from mom, because it's going to bind that fetal hemoglobin much more easily than the oxygen would bind adult hemoglobin. So the baby is definitely going to have a greater advantage in that oxygen binding because of the form of fetal hemoglobin, which is great because we want to make sure that our babies do have the ability to breathe, right? Because they're not using their lungs, so they're completely dependent upon mom for that oxygen saturation and that connection of the umbilical cord at the placenta to get that good healthy blood so that they can have that oxygen. Each red blood cell will contain about 280 million hemoglobin molecules, which means that each single red blood cell can carry over 1 billion molecules of oxygen. So that is a lot of oxygen inside of one little tiny red blood cell. In the peripheral capillaries where the oxygen is low, hemoglobin is going to release oxygen and it's going to pick up carbon dioxide. When this happens, now we have carbaminohemoglobin. So we have oxyhemoglobin if oxygen is bound, deoxyhemoglobin if oxygen dissociates and moves away from the hemoglobin molecule, and carbaminohemoglobin if that hemoglobin is binding carbon dioxide. It should bind the carbon dioxide because what we need to do is we need to move the carbon dioxide out of the system. So whenever the red blood cell releases the oxygen to the tissue, it will pick up the waste of carbon dioxide. The blood will continue on its pathway towards the lungs. So once it gets to the lungs, then the hemoglobin is going to bind up with oxygen and then release that CO2. So we'll breathe that CO2 out, we breathe oxygen in, so the hemoglobin will be picking up oxygen and again carrying it out to the tissues, dropping it off at the tissues, picking up the carbon dioxide, carrying the carbon dioxide to the lungs, dropping it off, picking up oxygen. It's a constant every single minute because that's about how long it'll take to go from the heart and through the rest of the system back up to the heart. It's about 60 seconds. Anemia is going to result when we have a hematocrit or hemoglobin content of the red blood cells being too low. Of course, this is going to interfere with the oxygen delivery to the tissues, so this can cause a number of different issues. So whenever we look at anemia, there are different types of anemia, and this all is about how are we interfering with tissue oxygen saturation. The red blood cell formation and turnover, basically 1% of those circulating red blood cells are going to be replaced every single day. That means you have about 2.5 to 3 million new red blood cells entering your bloodstream every second. And this equates to about 2 billion per day. That's normal. That's how it should be. So that's normal physiology. If it doesn't happen like that, then we could, of course, have issues with anemia. If we don't have enough red blood cells, if we don't have enough red blood cells, that means we won't have enough hemoglobin. If we don't have enough red blood cells, our hematocrit is going to be low as well. So whenever we're looking at thalassemia, this is an inherited genetic disorder that's caused, that should not say my, that should say by DNA mutations in the cells that make hemoglobin. So it will affect the oxygenation in the body. There are two types of thalassemia. So there's alpha thalassemia alpha, thalassemia beta. And this is all dependent upon which chain is affected in the hemoglobin structure, because remember, it was that quaternary structure that had 4 different chains, 2 alpha chains, 2 beta chains. So depending upon which chain is messed up will depend upon the severity of the thalassemia. If you have one mutated gene, then it's not going to be so bad, but if there are multiple mutated genes, then you're going to start experiencing more signs and symptoms. So a higher level of mutation in the genes is a higher level of severity of that thalassemia. Erythropoiesis is the term for red blood cell formation. Remember, we had hemopoiesis and hematopoiesis earlier on. That was formation of the formed elements, so as a whole. But if we're talking about just red blood cells, then the term is erythropoiesis. So in the embryos, the embryonic blood cells will move from the bloodstream to various organs like the liver, the spleen, the thymus, and the bone marrow, and then differentiate into stem cells. So the stem cells are then going to divide to help produce blood cells. In adults, this is going to occur only in what's called myeloid tissue, which is basically the red bone marrow. Now, in extreme cases of severe blood loss, it is possible for the yellow marrow to convert over to red marrow and actually produce blood cells, which is absolutely incredible that our body has the ability to do something like that. But in general, we're only going to be getting our red blood cell formation from our red bone marrow. Hemocytoblasts are also called hematopoietic stem cells, or HSCs. These are the stem cells in the myeloid tissue that are going to divide to produce either myeloid stem cells or lymphoid stem cells. So what you want to know about the difference between the two is the myeloid stem cells are going to become red blood cells and then 4 different types of white blood cells. The lymphoid stem cells are going to become exactly what their name sounds like, which is the lymphocytes. So just keep the lymph and the lymph together. Okay, lymphoid stem cells will only make lymphocytes, whereas the myeloid stem cells are going to make all of the red blood cells and then 4 types of the white blood cells. Hematologists are blood specialists who have identified several different stages of red blood cell maturation. So if you look over in the picture, what you can see on day 1, we have what's called pro-erythroblast. Remember, eventually when we have a red blood cell, this can also be called an erythrocyte. So a pro-erythroblast is the beginning stage of the differentiation for a red blood cell. Then we move into the erythroblasts. In days 2, 3, we've got a normoblast by day 4, and then a reticulocyte days 5 through 7. You can see here as the reticulocyte, there is no longer a nucleus within the cell. The reticulocyte is basically an immature red blood cell. So whenever you see that word reticulocyte, just think immature red blood cell. It's going to spend a few more days inside of the marrow until it is mature. Once it is mature, then it will come into the bloodstream and out of the marrow. Erythropoietin is a hormone that is going to stimulate erythropoiesis directly. Erythropoietin is going to be secreted by the kidneys and the liver whenever you have oxygen in the peripheral tissues as low. So we would call that hypoxia. Hypoxia is going to be the indication that it is time for erythropoietin to actually stimulate the process of erythropoiesis, which is creating more red blood cells. If we create more red blood cells, we have more hemoglobin. If we have more hemoglobin, we have more ability to bind and transport oxygen. So that's why low oxygen is going to be the stimulation for the hormone erythropoietin. Now, under maximum EPO stimulation, the red bone marrow can increase the RBC formation tenfold to about 30 million cells per second. Indirect stimulation of erythropoiesis is going to be done by the thyroid hormone thyroxine, by the male steroid hormone androgens, and then also by growth hormone. So if you are a male, then you have a higher ability to have an indirect stimulation of erythropoiesis. If you are working out, doing things like bursting exercises and therefore releasing that growth hormone, then you also would have a greater advantage of stimulating that erythropoiesis, because obviously in working out, you're conditioning and training your body. It's going to need more oxygen, and so that erythropoietin will be stimulated to help you create more red blood cells and have more oxygen. Blood doping is a process that is used by some athletes. So what they do is they will re-infuse packed red blood cells in order to elevate their hematocrit levels. By doing this, they then get more oxygen delivery to the muscles, and of course, if you have more oxygen, that means that you can go longer, you can go harder, you can go faster. So therefore, they have a competitive advantage in their performance because they have more oxygen delivery to those muscles that they need to use. Unfortunately, this will make the blood more viscous, and so therefore it will increase the workload on the heart. When you think about viscosity, think about the difference between water versus honey or maple syrup. So whenever the blood becomes more viscous, then that means we are going to have problems with flow. If we have problems with flow, then of course this is going to stress the heart out, and so therefore the blood doping can become a dangerous issue. Most people know about Lance Armstrong because he is of course the poster child for what happens whenever you are caught with doing something like this. So unfortunate situation, but we all know the reality of what occurred. Now at this point, erythropoiesis, however, does require amino acids, iron, folic acid, and then vitamins B12 and B6. So even if you have low oxygen levels and you're stimulating that erythropoietin, which is going to kick in the process of erythropoiesis, you're still going to need these various factors to help with building up these new red blood cells. If you do not have them, of course, then there is going to be a deficiency in that erythropoietic process. A lack of vitamin B12, just that one alone, can lead to something that's called pernicious anemia. Pernicious anemia basically means the body cannot make enough red blood cells because there's not enough vitamin B12. Now, I'll put an asterisk right there. The IF stands for intrinsic factor, and whenever we get into the digestive system, we'll talk about intrinsic factor and how important it is to being able to absorb B12. So you may not have enough B12, or you may have enough B12 but you don't have enough intrinsic factor to absorb the B12 that you do have. So again, everything is connected. You can't just take vitamins willy-nilly and think, oh, well, I need this for that and this for that, and you've got like 13 different vitamins that you take for all single individual things, when in reality, the whole system is going to have to work together as a unit. So even if you're taking the vitamin B12, I know people sometimes go get the sublingual drops, and sometimes people get vitamin B12 shots. If your digestive system is not working appropriately and you don't have the intrinsic factor to absorb it, you are going to basically be on a spinning hamster wheel. You're not going to get anywhere, even though you feel like you're trying. Now, whenever it comes to the hemoglobin, it is going to be recycled, and what's going to happen is we will have these cells within the system called macrophages in various places like the spleen, the liver, the red bone marrow, and these macrophages are actually going to engulf the aged red blood cells. Remember, they only live for about 120 days. So whenever the macrophages engulf these, they will remove the hemoglobin molecules from these ruptured red blood cells and then break the hemoglobin into components. So the alpha and the beta chains of the hemoglobin are going to be filtered by the kidneys and then eliminated in the urine, but highlight this, the iron of each heme unit is going to be recycled. We are going to keep the iron because it is going to be, of course, that integral component of that hemoglobin. Looking at this chart here, what you can see is the macrophage is in purple. Okay, so you've got the macrophages in various locations. When you think about the macrophage, this is basically considered a phagocytic cell. Phagocytic starting with a P. Okay, think about Pac-Man. Pac-Man starts with a P. So phagocytic cells eat things, just like Pac-Man goes all around the screen eating all of those dots. So a macrophage is going to basically eat these red blood cells that are old, or you can see that's about 90%, or 10% would be the damaged red blood cells. So if something in the bloodstream actually ruptures, if the red blood cell ruptures, then the macrophage can go over and eat up that mess. Okay? So once it eats it, then it's going to start breaking that heme unit apart. As you can see in the purple arrow, the iron is going to come out, and we're going to transport the iron in the bloodstream by transferrin. So that's a transport protein. We're going to go put it over into the bone because those developing red blood cells are going to absorb the amino acids in the iron from the bloodstream in order to synthesize new hemoglobin molecules. So once we've got that new hemoglobin molecule and that red blood cell has matured, then that new red blood cell can be released into the bloodstream and again live for about 120 days. Once it gets to its prime and it's over, it's too old to do anything anymore, again the macrophage is going to eat it, do the same thing, break apart the heme, recycle the iron. Now the other thing that you see happening is in green. The heme is ultimately going to be converted to biliverdin, which is then going to be converted to bilirubin. So bilirubin is going to be bound to albumin, one of those transport proteins that we talked about earlier. It's going to be bound to albumin in the bloodstream in order to move over into the liver. I have a little arrow there for you. Bilirubin, if there is too much buildup of bilirubin, then we are going to get jaundice. Okay, so bilirubin is going to have an orangey yellowish color to it, and if you have an accumulation of that bilirubin within the liver and it can't get everything out within a specific amount of time, if there is an accumulation and a buildup, then things will start turning yellow, like the skin will turn yellow, the whites of the eyes start turning yellow. If you have jaundice, then that is a sign that we have a problem within the liver, and that definitely needs to be evaluated. You know, a lot of newborns will sometimes have jaundice, and this is because as a newborn, a lot of their red blood cells are breaking down and transitioning as they're going from that fetal hemoglobin to that adult hemoglobin. So if there is a very quick breakdown of all of those red blood cells, then it is possible for that newborn to get jaundice because their liver just isn't as good at processing things yet because, again, they're little bitty tiny babies. So normally you give them a few days and everything is fine and everything gets cleared up as that liver learns how to work and move things out of the system appropriately. But in an adult, we need to look and see what's going on inside of the liver. Is there a blockage to one of the ducts or whatever it is? So what should happen is bilirubin should be excreted in the bile. If bilirubin is excreted in the bile, then it is going to get into the large intestine and ultimately will be converted to urobilin and stercobilin, and that is going to be eliminated in the feces. Some of the urobilin can come out of the large intestine, get absorbed into the bloodstream, and go over into the kidney, And as you can see, that urobilin can then be eliminated in the urine. So the urobilin is part of the reason why you will have yellow pee, okay? Because the kidneys are going to excrete some of that hemoglobin, but also that urobilin, which is going to give it that yellow color. You can see that within the large intestine, the bacteria that convert the bilirubin to urobilin and stercobilin, again, you're going to have that color because of the presence of those two things in varying proportions. So the more urobilin, the more yellow it's going to be, the more stercobilin, then the more darker brown it's going to be. If you want a little video about this hemoglobin recycling, then here is a little link for a YouTube video for you right here. These slides are the word slides, basically everything that I just talked about in the picture, but if you want to go through and read about the hemoglobin recycling in these word slides, then there it is for you. Same thing here, except for at the bottom. So I didn't talk about what's at the bottom. If we have the excess transferrins removed in the liver and the spleen, then we can store iron in other storage forms called ferritin and hemosiderin. If there is too much iron, then this is called hemochromatosis. Hemochromatosis is iron overload. It is very often a genetic issue, but it can cause some serious damage to your body, including to the heart, the liver, and the pancreas. People who have the familial hemochromatosis, where this is a genetic condition, will often go give blood in order to help draw some of that iron out of their system. Hemoglobinuria is where we have red or brown urine. This is due to abnormally high hemolysis. Remember, lysis means to cut. So if we're having the red blood cells basically being cut up or torn apart, then that is what we would say is hemolysis within the bloodstream. Too much of that going on could then lead to changes in the color of the urine, which is definitely something that needs to be looked at immediately. And hematuria is whole red blood cells in the urine. Now, this can be due to the kidneys or the blood vessel being damaged. So the red blood cells aren't necessarily rupturing, but instead they are actually coming out whole. So that means that somewhere there is a leak that's allowing the red blood cells to come out and end up in the urine. You do have to be cautious with diagnosing hematuria because it is possible, if it is a female, that this may not actually be damaged somewhere, but it could actually be menstrual blood. So just keep in mind that if she is on her cycle, of course you want to ask, make sure that you evaluate that appropriately. So in order to help you keep some of your terms straight, you can see here that whenever we do blood labs, we've already talked about the hematocrit being the percentage of the formed elements in the whole blood, higher for males than it is for females. If the hematocrit is too high, we could call this polycythemia. If it's too low, we would say anemia. And as you can see there, there's a number of different other tests and values that you can look at to see what's going on inside of the body, and then there are terms that we would associate with those abnormal values as well. When it comes to blood typing, what we're going to be looking at are these antigens that are actually going to help identify what type of red blood cell you have. So surface antigens are the substances on the actual membrane that will identify the cells to the immune system. Your immune system will know self versus non-self based upon the antigens that are presented on cellular membranes. So of course it will ignore the normal cells and then go and attack anything that does not match your specific antigen. Now, when it comes to blood typing, there are a number of different antigens that can be found on red blood cells, but we're going to talk about the most common ones, which would be A, B, and then RH, or sometimes referred to as D. Some people follow a blood type diet, not because of a fad thing, but if you look further into the blood type diet, it's all about what are the antigens on that particular red blood cell and how does it interact with the antigens that are on specific types of foods. So it's really more so about getting your system to not freak out when different foods are introduced into the body because they have the same or similar type antigens to what are being presented on your blood cells. Some people love the blood type diet. Other people have tried it. They don't really see that much of a difference. So it is variable for each individual. But if somebody is struggling with something and they just can't quite get things correct and they don't really know what to eat or how to eat, it is at least a good place to start. And if they find it to be beneficial, then great. And if not, then time to be a detective and try something else. So we're going to talk about the 4 major blood types. We have type A, B, AB, and O. Whenever we're looking at type A, then that basically means that it has the surface antigen of A. And then for B, surface antigen B. The weird part is where we get to type O, because it doesn't have either one. It doesn't have A or B. So think about type O. It looks like a big 0, okay? So it's not going to have an antigen on its plasma membrane. For the Rh blood group, this is going to be based on either the presence or the absence of the Rh antigen. Obviously, if it's present, then we're going to say that this is an Rh positive. It is present, it is there. If it's Rh negative, then that means it is absent, it is not there. So we would have type A positive, meaning surface antigen A, and we have the presence of the Rh antigen. Type A negative means we have surface antigen A only. There is no Rh antigen, and so on and so forth with the other blood types. As we're looking at the surface antigens on the red blood cell, we can call these things agglutinogens. So think about that last 4 letters in that word, gens. Agglutinogens, antigens. So we're talking about the same thing, okay? Agglutinogens are the surface antigens on the red blood cell. This is what is going to determine your blood type. Do you have A? Do you have B? Do you have both, or do you have none? The agglutinogens are going to tell us what your blood type is. This is what the immune system is going to be aware of so that it knows what is you and then what is something else. Agglutinins are not the same as agglutinogens. Agglutinins are the actual antibodies in the plasma. Remember, the plasma, 90% of it is water. Remember, it goes to the top whenever you centrifuge a blood sample, so it floats up. The plasma is 55% of the blood. The formed elements are 45% of the blood. Remember, the red blood cells sink all the way to the bottom. So you would find the agglutinogens on the actual red blood cells, but you find the agglutinins within the plasma, okay? So the antibodies in the plasma are the agglutinins, and these will attack the antigens on any foreign red blood cells, and that will then cause something called agglutination, which basically means clumping, and it will cause this clump, which means it will send a signal to the immune system saying, we have a problem. Come and look at what's going on over here. So the clumping is not going to be a good thing, because then this is going to cause problems within the blood. This is why it's very important to make sure that whenever you're giving someone blood, they have to have have evaluated their blood type so that you know what it is and what you can give without creating an immune reaction and this agglutination. Obviously, if it's an emergency, then you may not have time to determine their blood type, in which case we'll talk about what you do in those situations in a minute. So, because the agglutinins are antibodies, that means that type A blood in its plasma is going to have anti-B antibodies. It's going to say, do not give me any B blood. I don't like B. I am anti-B. Okay? Type B is, of course, going to say, don't give me any A blood. I do not like A. I am anti-A. Type O blood is going to have both anti-A and anti-B antibodies. Now remember, type O blood had 0 antigens on the plasma membrane, but it has both of these antibodies inside of the plasma itself. Type AB blood has neither anti-A nor anti-B antibodies. And if we're talking about the Rh factor, only sensitized Rh negative blood has the anti-Rh antibodies. I'll talk about how it gets sensitized here in a moment. So looking at this chart, I really like this slide because this chart will basically help lay out for you what type of surface antigen is actually on the cell itself, and then what type of antibody is going to be floating around in the plasma. So this is a good breakdown of all of the 4 different ones where you can see their antigens and their antibodies. As you look at the surface antigen A, you see it looks like a little red circle. In surface antigen B, they have drawn it into a blue triangle. So on your type AB blood, you can see that it has both. But remember, type AB blood has no antibody against anti-A or anti-B. Type O blood, remember, no antigen, 0 antigen on the surface of the plasma membrane, but it does have both anti-A and anti-B antibodies. So, when we're talking about blood donation and giving someone a transfusion reaction can occur if the donor and the recipient blood types are not compatible. See, it doesn't say that it has to specifically and exactly match, like A plus can only have A plus. It's not the way it is. It's not a specific and exact match. It just has to be compatible. So what that means is we can give different types of blood. We just have to know who is compatible with who. So the plasma antibody will meet its specific surface antigen, and if it is not compatible, then those red blood cells will agglutinate or clump, and then they can break apart, as you see in the picture there. So clumps and fragments of red blood cells under attack form these drifting masses, and they can then plug the small blood vessels that are within various organs and then destroy those particular tissues. So this is why it can become a system-wide problem if you give someone the wrong blood that is not compatible with theirs. In our little picture example, you can see here we've got the little blue triangles. So that means that this is a person who has type B blood. However, what you can see here is that in this reaction, it has come across anti-B antibodies. Those anti-B antibodies don't like the B antigens, so they're going to go attack those B antigens. And whenever they do the attack, you can see here that everything is going to start to clump together, and then the red blood cells are going to hemolyze or rupture. So, not a good thing. Compatibility testing will be performed in advance of transfusions, and the cross-match testing will reveal cross-reactions between the donor's red blood cells and the recipient's plasma. Type O negative is the universal donor. It's possible for cross-reactions to still occur because there are more surface antigens other than the A and the B, but in general, in emergency situations, when you don't have time to do compatibility testing, if the person is bleeding out in the emergency department, type O negative is what is going to be given because it is least likely to cause a problem. So if we're going to evaluate and try to do some compatibility testing here, we're going to be looking to see what's going on whenever we have anti-A and anti-B put on different blood samples, and then of course anti-RH as well. So on this picture, what you can see is the anti-A caused clumping. That means that there are A antigens on that red blood cell surface in that sample. So if anti-A comes against it and causes the clumping, we know it is A blood. You see anti-B went against it and there was no clumping, so there was no reaction because there are A antigens there. And A antigens are okay with anti-B because that is what is in the plasma of that particular blood type. On the anti-RH, you can see that that caused clumping. So since that caused clumping, then that means that we know that the RH is present. That RH antigen is there. So therefore, the clumping on anti-A and the clumping on anti-RH means we have a blood type of A positive. And then that's how you would figure it out in regards to compatibility testing. So again, with the next one, you can see clumping on the anti-B. That means we have B blood. Clumping on the anti-RH, that means it's positive. Clumping on the next one in anti-A and anti-B and anti-RH. So clumping, clumping, clumping means we have A, B positive. And then on the very last one, no clumping, no clumping, no clumping, that means that there is no antigens, and any of these, since they did not clump, that means that we have an O negative blood type. So most people will use this little cheat sheet to know, okay, if I'm looking at the left-hand side of the page, I can see that this is the column for the recipient's blood. So if the recipient has O negative, then what could I give that recipient? Who is an acceptable donor? And as you can see, the green check mark says the only acceptable donor for an O negative recipient is an O negative donor. No one else is going to work. Any other blood type that you give to an O negative person is going to cause clumping, and you will have that transfusion reaction, which can lead to some severe issues. So we don't want to do that. Okay? So this is one of the reasons why I harp on you guys to make sure you're paying attention, reading, looking, listening, because you can see here, if you just barely mess up and you give that person O positive because you missed one little small thing, the positive sign versus the negative sign, then you could have potentially just killed that person. So it is that big of a deal to make sure you're paying attention whenever you are in the medical field. If you look at the recipient all the way at the bottom, the AB plus, so AB positive recipient, you can see that all the way across the bottom, every single type of blood can donate to an AB positive recipient. So whenever we're looking at an AB positive person coming in needing blood, that one is very easy because that person can take anything from anyone. This used to be called the universal recipient blood type, although that terminology is not used as much anymore. But the other thing I want to show you is look at the O negative in regards to the donor column. The O negative in the donor column can give to every single type of recipient. So for that reason, we will call O negative the universal donor. So just remember, donor has an O in it. The universal donor is O negative. It can give to anyone without there being this transfusion reaction. Well, I should say most. It can give to most without there being a transfusion reaction, which is why it is the one that is most commonly used inside of the emergency department. And this is why if you have O negative blood, then people will definitely want you to donate so that way they can have plenty on stock for emergency situations. Now, let's talk about that Rh factor that could become a problem if that blood is sensitized in an Rh negative person. So the example here is going to be what's called hemolytic disease of the newborn. Whenever you're looking at this picture, what you can see is this is a mother who is pregnant for the first time. And she is Rh negative, but her baby is Rh positive. Okay? In the first pregnancy, it's generally not a big deal because of the separation between the maternal blood and the fetal blood. So they generally can stay pretty separate. A few of the fetal cells are going to enter the maternal bloodstream, but the immune system isn't going to go crazy with it. So whenever we're looking at pregnancy number 1, no big deal. However, once the delivery occurs, there's going to be some mixing of the blood. So you can see here that whenever that positive blood gets over into mom's bloodstream, what it can do is it can sensitize that Rh negative blood, and the immune system can kind of set up a reaction to say, okay, the next time we see this, we're going to go after this. So it kind of learns that Rh positive blood type is not of self, and it's ready the next time that Rh positive blood type comes around. So it's important that if a mom is Rh negative with an Rh positive baby, if she's going to have more pregnancies later on, she needs to make sure she is verbal with her doctor about how she had an Rh positive baby with her first child, because they will want to make sure to check if this next child is also Rh positive, because there's going to be something that's going to have to be done in order to protect mom and the baby. So if we have the sensitization here at delivery, then you can see here that the maternal antibodies to the Rh antigen can be built up. So about 20% of the Rh negative mothers who carry Rh positive children will become sensitized within 6 months of delivery. Now, these antibodies are not going to be produced in significant amounts, so that's why it's not going to affect baby number 1, because baby number 1 is already out of the body. The problem can arise with baby number 2 or baby number 3. So what you can see here is if the maternal anti-Rh antibodies produced after the first delivery cross the placenta and enter the fetal bloodstream, those antibodies can destroy the fetal red blood cells, producing a dangerous anemia. So the fetal demand for blood cells will increase. They leave the bone marrow and enter the bloodstream before completing their development. Now, these immature red blood cells are erythroblasts, so hemolytic disease of the newborn is also known as erythroblastosis fetalis. If there is no treatment, the fetus may die before delivery or shortly thereafter. A newborn with severe HDN is anemic, and the high concentration of circulating bilirubin will produce jaundice. Because those maternal antibodies will remain active in the newborn for 1 to 2 months after delivery, the infant's entire blood volume may require replacement to remove the maternal anti-Rh antibodies as well as the damaged red blood cells. Fortunately, the mother's anti-Rh antibody production can be prevented if such antibodies, available under the name of RhoGAM, are administered to the mother in weeks 26 through 28 of pregnancy and during and after delivery. So the RhoGAM shot is the reason why moms who are Rh negative that first had an Rh positive baby will want to talk to their doctor, because if the RhoGAM shot is given, then this hemolytic disease of the newborn can be prevented. So the RhoGAM has pretty much helped eliminate HDN within the United States. So it's really nothing to necessarily be scared of if you are a mom who is negative with a first baby who is positive, because it's something that can easily be treated. My case, exactly, my first baby was Rh positive and I'm Rh negative. So I knew about the RhoGAM, I knew what I needed to say about successive pregnancies, and had no problems with my second or third child. Okay, so now let's talk about white blood cells. These are also called leukocytes. The leukocytes, remember, are not as numerous as the red blood cells, and they look different as well. They are not going to be biconcave disc. Instead, they are going to have cells that are much larger. They are going to have nuclei. They will not have hemoglobin. And remember, you want to associate these with the immune system because they're going to help defend the body. Most white blood cells are in connective tissue proper and organs of the lymphatic system. A very small fraction of the white blood cells will circulate in the blood, which is why you will have a harder time finding these cells on the blood slides. When we're looking at the characteristics of the circulating white blood cells, these do have the ability to migrate out of the bloodstream. That's called diapedesis. So basically they squeeze through the endothelial cells of the vessels. They are also capable of what's called amoeboid movement. And if you look at that particular video, that is what amoeboid movement looks like. So what they're doing is moving the cytoplasm around in order to move somewhere else in the body. So it's like these cytoplasmic extensions just reach out and then just kind of move wherever they want to go. The white blood cells are attracted to specific chemical stimuli. We call this positive chemotaxis. So whenever we say chemotaxis, think about a cell phone. Basically, a chemical cell phone signal that's calling the white blood cell, telling them, hey, I want you to come over here because we have an issue over here. Some white blood cells are phagocytic. Remember, when we think phagocytic, think about Pac-Man. It means they're going to eat or engulf something. The differential count of the white blood cell population means that we can detect infection or inflammation or allergic reactions because we're going to break down the various different types of white blood cells into how many are there actually within the sample. That's a differential count. We separate out how many neutrophils, how many eosinophils, how many basophils, etc. because each one of those white blood cells is going to have their own particular function. So if we see a high or low count on the different types of white blood cells, then we can kind of go and figure out what could potentially be going on. There are a number of terms that you will want to remember about white blood cell disorders. So leukopenia is a low white blood cell count, whereas leukocytosis is a high white blood cell count. Leukemia is cancer of white blood cells that is indicated by extreme leukocytosis. For our differential count, for our different types of white blood cells, You have this mnemonic here that I still use to this day, never let monkeys eat bananas. That stands for the most to least percentage in the white blood cell diff count. So neutrophils is the never, lymphocytes is let, monocytes is monkeys, eosinophils is eat, and then basophils is bananas. So NLMEB, never let monkeys eat bananas, is a very quick way to look at labs whenever they come out, and you can see, should I say that there is a problem here if I have more monocytes than I actually have neutrophils? Yes, absolutely, we have an issue, because it should be neutrophils, lymphocytes, monocytes, eosinophils, and basophils from most to least. When things start getting out of order, then we have an issue. And of course, they may not be out of order, but you could have a higher neutrophil count, and that would generally be flagged on the labs as well. Maybe a lower count, that could be flagged. So whenever we talk about what each one is for specifically, then you will know maybe what could be the problem in that particular person. You have pictures there of all of the different types of white blood cells. As you can see, they all have quite large nuclei. Some have more than one nuclei, so multiple nucleuses within a single cell. And then like the lymphocyte, very easy to identify because the nucleus basically takes up the entire cell. It looks like there's just a small little halo around the nucleus of a lymphocyte. So leukopoiesis is the term for white blood cell production. Remember, erythropoiesis is red blood cell production. Hematopoiesis or hemopoiesis is production of formed elements. So lots of different poiesis here. Hemocytoblasts will produce those myeloid stem cells and the lymphoid stem cells like we talked about earlier. The myeloid stem cells will divide to produce progenitor cells, and these will give rise to all of the formed elements except for the lymphocytes. Remember, the lymphocytes come from lymphocytopoiesis because they are from the lymphoid stem cells, not the myeloid stem cells. Neutrophils are also called polymorphonuclear leukocytes. Highest number that you should see on the diff counts, this should be 50 to 70% of the circulating white blood cells. They are pale cytoplasmic granules, and they contain lysosomal enzymes and bactericidal compounds like the hydrogen peroxide, which enable them to kill pathogens. Neutrophils are very active phagocytic cells. They will attack and digest bacteria. Bacteria is what you really want to think about whenever it comes to neutrophils, because that is what they are there for. Degranulation is the reduction in the number of cytoplasmic granules that will occur when the vesicles containing the pathogen fuse with the lysosomes containing enzymes and defensins, which are basically going to destroy the bacteria. So they will release prostaglandins, which will increase inflammation, and leukotrienes, which are going to be the signal to other white blood cells that they are calling out for help. This is important because the neutrophils are very, very important in helping with digesting and killing bacteria, but they do not live for very long. They're only going to live for about 10 hours or so. So they're going to do a great job of helping to clean all of the bacteria up, but they have their limitations. So other things are going to have to be called in to help with this process as well. Dead neutrophils are what help contribute to pus. So whenever they've eaten all of that bacteria and then they die, if you've ever squeezed out a pimple and you've seen the pus come out, then you're looking at multiple different cell fragments of those neutrophils as well as that bacteria. Eosinophils are also called acidophils. 2 to 4% of the circulating white blood cells. They will engulf bacteria, protozoa, and cellular debris. But the important thing to remember about the eosinophils are the two words in red, which is parasites and allergens. So the eosinophils have the ability to attack large parasites by releasing toxic compounds like nitric oxide and cytotoxic enzymes. Because these parasites can very often be bigger than what a cell could actually engulf or ingest, the eosinophils will release these different compounds in order to basically break down those parasites since they can't be engulfed by something else. The eosinophils are also going to be associated with allergies, and they will release enzymes that help reduce inflammation caused by the mast cells and the neutrophils. Basophils are less than 1%, so this is your very end. This is the bananas. Never let monkeys eat bananas. The basophils, very small percentage. When you think about these, think about histamine and heparin, because these are the two things that the basophils are going to release. The histamine, which will dilate the blood vessels and cause swelling, and the heparin, which will prevent blood clotting. Monocytes are large cells, very large. This is 2 to 8% of the circulating white blood cells, and they remain in the bloodstream for about 24 hours. Then they will enter the peripheral tissues to become macrophages. So circle that word of macrophages. Monocytes in the blood, macrophages in the tissues. Remember the macrophages, the P, Pac-Man, they are phagocytes, and they will eat up these pathogens. They will also release chemicals that will attract other phagocytic cells, remember positive chemotaxis, and then fibroblasts to that particular area of injury in order to start laying down scar tissue. Lymphocytes are going to continuously migrate in and out of the bloodstream. They will mostly be in the organs of the lymphatic system. So lymphocytes come from lymphoid stem cells through the process of lymphocytopoiesis, and they live in the lymphatic system. So all of the lymph goes together. Everything with lymph stays together. This is going to be part of the body's specific defense system. We'll talk about it more when we get into the immune system, but for right now we will talk about the 3 different types of lymphocytes. Your 3 types include your T cells, which is all about cell-mediated immunity. That means it can attack foreign cells or even control other lymphocytes. So T lymphocytes, think cells. B lymphocytes, think plasma. So this is not cell-mediated immunity, this is humoral immunity, which means it will differentiate into plasma cells, which helps synthesize the antibodies. So T is for the cell, B is for the plasma. And then natural killer cells are going to detect and destroy abnormal cells. Again, we'll talk about each one of these more as we get into the immune system. White blood cell development, so we have some lymphoid stem cells that will remain in the red bone marrow that differentiate into B cells or natural killer cells. Others will migrate from the red bone marrow to the peripheral lymphatic tissues. The thymus is very important, incredibly important. This is the organ that sits in the chest near the heart, and it is the one that is going to be producing our T cells. The thymus does atrophy with age, so that is why it is more common for people who are elderly to become ill a whole lot easier because of the atrophy of the thymus and the reduction in the T cell population. Colony stimulating factors are hormones that will regulate white blood cell population. So this is not just about what's going on in the bone marrow, this is also about a hormonal control as well. Multi-CSF will accelerate the production of the granulocytes, monocytes, platelets, and the red blood cells. GM-CSF will stimulate granulocyte and monocyte production. G-CSF will stimulate the granulocyte production, and then the M-CSF will stimulate monocyte production. So What you see on this picture here is we're starting with a hemocytoblast or a hematopoietic stem cell. Remember, we have two different divisions, either myeloid or lymphoid. Remember, the only thing we're going to be getting from lymphoid is going to be the lymphocyte. So that is going to be in the lymphatic system. From the myeloid, this is where we're going to get our red blood cells, our platelets, and our other 4 types of white blood cells. So what you see on this chart is that you're going to need the hormone EPO to help with the production of the red blood cells. You have various hormones like multi-CSF, GM-CSF, G-CSF, and MCSF to help with the production of your white blood cells. Remember, the platelets are not actual cells themselves, they are just cell fragments. So what you see on that picture there is the megakaryocyte is the cell that is going to fragment and separate out in order to give us our platelets. This is why the platelets look so small on the microscope slides in lab. They look like little specks of dirt because they're not actually whole cells themselves, they're just little pieces of cells. Across the bottom, for the white blood cells, what you see is the basophils, eosinophils, and neutrophils are all classified as granulocytes because you can see all of those fine little granules whenever you look at them underneath the microscope. And then the monocytes and lymphocytes are categorized as agranulocytes because you don't see that as well on the microscope when you are looking at those 2 types of white blood cells. And finally, the platelets, aka the thrombocytes. So again, these are the cell fragments that are going to help with clotting. They circulate for 9 to 12 days and then get removed by phagocytes, mainly in the spleen. 150,000 to 500,000 per microliter of blood, and 1/3 of those platelets are going to, in the body, are stored in the organs like the spleen, but they will be mobilized during a circulatory crisis. So they're not just going to be floating around in the blood all of the time. They are actually going to be in storage, and then whenever you massively cut your finger with that kitchen knife as you're trying to cook dinner, then they're going to come out of the spleen and head towards your finger so that they can begin the process of the clotting so you don't bleed out. Of course, the platelets, yes, they are going to be for clotting, so that means they will release important clotting chemicals. They will temporarily patch the damaged vessel wall and then reduce the size of the break in the vessel wall. There is a little video for you there if you want to watch it about how the platelets are going to help with the clotting process. Thrombocytopoiesis is platelet production. It occurs in the red bone marrow. Remember, we also have, again, leukocytopoiesis, erythropoiesis, and then we also have hematopoiesis or hemopoiesis. So, as I told you at the very beginning of this, lots of terminology, lots of things to keep straight, lots of things to remember. Again, the megakaryocytes that you just saw on the picture before are going to be the huge cells that are ultimately going to produce the platelets whenever they shed their cytoplasm. And there is hormonal control of platelet production as well. This includes TPO, IL-6, and multi-CSF. So hormones are going to be involved in red blood cells, white blood cells, and platelet production. Hemostasis is what we call the cessation of bleeding. It has 3 different phases: the vascular phase, the platelet phase, and the coagulation phase. These get followed up by clot retraction and then eventually fibrinolysis, which is just basically breaking down the fibrin meshwork that helped create the clot and then dissolving it so that it does not stay there. So that is the normal physiological process. You create a blood clot and then ultimately you clean up the blood clot. It's not going to stay around for forever. So I have this video for you here, which is going to cover the remaining slides. That way you can actually see this in a 3D version, which I think is much better than just looking at pictures and reading words on the slides. So any of the remaining slides, for those of you who like words, you can feel free to use that. Otherwise, watch that video, and that will cover the end of the chapter for blood.